Hello, and welcome to Chapter 2. So we continue our discussion. We're building into transmission genetics. And in order to really understand how traits and genes are passed on from generation to generation, we first need to understand the concept of cell division and what happens in each case, both mitosis and meiosis. So there's every living thing contains a substance described as the genetic material, and we know from last chapter that that is our DNA. The genetic material is not protein; it's DNA. So an organism's DNA that includes its array of genes is organized on chromosomes. And the chromosomes are the vehicle for transmitting the genetic information. And the manner in which the chromosomes are generated or transmitted from one generation of cells to the next and from one organism to their descendants, we need the first, it's a very precise way of doing it. So that's what we're going to look at here. We're going to look at the two ways or the two processes for distributing those chromosomes, and from cell to cell, as well as from the within an organism, from parent to children too. So our next slide here is our learning objectives. I won't read them over. You could take a look at those. So again, every living organism has genetic material, and it's composed of nucleic acid, your deoxyribonucleic acid, and organized on chromosomes. So in eukaryotes, transmission of genetic material is through the chromosomes. And the two processes that the chromosomes are transmitted is mitosis and meiosis. So meiosis is the production of the sex cells, which are the gametes or spores, and mitosis leads to the production of two cells, each with the same number of chromosomes as the parent cell. So there are some divisional phases where the cells divide, but then there's also non-divisional phases. And during those phases, chromosomes uncoil into a pretty scattered network within the nucleus, and that they're called chromatin at that point. So chromatin, what that really is whenever the cells are not undergoing division, our genetic material that constitutes the chromosomes, instead of being tightly wound and organized, it basically unfolds and uncoils and spreads out within the nucleus, and that's really what chromatin really is. So we're going to go on now and look at our definitions first of our mitosis and meiosis. So mitosis, the chromosomes are copied and distributed, and it gives you results in two daughter cells, each receiving a diploid set. So our N number is 23, so we end up with a full diploid amount of 46 total chromosomes. And then meiosis is where chromosomes are copied and distributed, but now the result is the gametes receive only half the number of chromosomes. So in meiosis, the result is a haploid number, and that's the 23 chromosomes, the 23 individual chromosomes, not 23 pairs. So in mitosis, it's 23 pairs for a total of 46 chromosomes. Meiosis, it's one pair, or like one chromosome from each pair, and you wind up with a haploid number, or half the number of chromosomes. You have your 23 in meiosis. So there's two main types of cells, prokaryotic and eukaryote. So prokaryotic is it just has a nucleoid, not a nucleus, and that's usually our bacteria and some algae, archaea, and then eukaryotic cells are the protists, plants, fungi, and animals. And they have a nucleus. And but all of the cells share common features, namely they're enveloped by a plasma membrane. That's the outer barrier. They contain the DNA, and we know that resides in our nucleus. And they have ribosomes, and then a variety of other organelles. So here's that cell, what that looks like. So we can see the plasma membrane. That's plasma membrane right over here. So it's really this blue barrier right here. It's a bilipid layer. The line points right to the side, but it's not pointing to the inside. It's literally this, let me get my little marker here. It's literally this part right there, that separation. So that's actually the reason is the plasma membrane itself is it's a double layer membrane. So it's often referred to as a bilayer phosphate or phospholipid, bilayer phospholipid membrane. And so the lipids point inward. Oftentimes you look at it this way, kind of thing, and you have the head and the tail. And then this is basically the structure here that we're looking at right there. And the reason is it allows for permeability. So we see here some of the indentions are engulfing, but also the way this is made up is to look at that exterior part as a semi-permeable barrier. It doesn't, like, it's semi-permeable because it doesn't let everything into the cell, nor does it keep everything trapped in. Certain things cross that bilayer or plasma membrane. So, and we know that nutrients, or one of them, they get broken down, and then sugars cross over. But then we see waste products transferring in and out. So the plasma membrane is semi-permeable. Some things are allowed in, others are not. Often depends on the size. And then there's a few mechanisms like osmosis and diffusion, active and passive transport, a variety of ways that that material can pass in and out of the cell. And then so that's the membrane. And then the purple right in here is the cytoplasm. And then a bunch of, here's the Golgi bodies right here. And then the ribosomes are, let me move up a little bit. So we have the ribosomes over here, and they're bound ribosomes on some of them, tightly wound. And this is the rough endoplasmic reticulum that has all the little bumps on it. But then, so they're bound, back to the bound ribosomes. Those ribosomes are bound to the endoplasmic reticulum. And it's the rough endoplasmic reticulum is the one that has the bound ribosomes on it. But then down here we see free ribosomes down here. They're not bound. And we see smooth endoplasmic reticulum. So the smooth endoplasmic reticulum, or smooth ER, does not have ribosomes on them. The rough ER has the ribosomes bound to them. And then we see the lysosomes right here. We see the nucleus right in here. And then the chromatin at this point. Only chromatin's really not visible. The chromosomes are really only visible under mitosis and meiosis. But we see the nucleolus in the center right there, in the chromatin, and then the nuclear envelope also has a membrane that keeps the nucleus tightly bound. So that's our cell structure right there. And we're going to look at a little bit more detail into each of them. So the plasma membrane, it surrounds all cells. It's permeable. It's a double layer or bilayer of phospholipids. Hydrophilic and hydrophobic ends, so some of them want to let in water, others not. So the permeability, again, we covered that. And it's a key thing here is that it's a delimiter from the external environment, so it's a protective barrier too. So the nucleus is found in eukaryotes, and it's membrane bound. We saw that wrapper around it. And it houses, the nucleus is where all your DNA is located. The nucleolus is further buried inside, or like the center of the nucleus, where the ribosomal RNA is synthesized. And the nucleolus is also has an organizer region where the DNA encodes the RNA, or ribosomal RNA. And then the nucleoid is in prokaryotes, and it's not membrane bound. We're not going to go into a lot of detail on prokaryotes. We're just going to focus on the eukaryotic cell. And then that cytoplasm is kind of like the glue or the gel or gelatinous environment that keeps all the organelles in place within the cell. The cytosol is a colloidal material that also surrounds the cells. And then the cytoskeleton is similar to your own skeleton in that it provides a framework for your body. So the cytoskeleton is a system of tubules and filaments that give structure, that give a structural function to maintain the shape of the cell, and it also has functions for transporting material within the cell. And that transport is carried out by motor proteins on the surface of the tubules that provide the structure. And then microfilaments are also a part of the cytoskeleton, and they're composed of actin, and its association with myosin results in muscle contraction. So that's more on a macroscopic level. You think of your muscle, your biceps. When you think of that, your biceps are made of muscle cells. A collection of cells is a tissue, and a collection of tissues is an organ, and a collection of organs is an organ system. So your muscles are an organ, and the myofilaments, right down to the microscopic level of the cell, are composed of actin, and then when they associate with myosin, together, when they all associate together, you get a muscle contraction. Is one example of where they're related. Then our endoplasmic reticulum is an organelle, much like everything inside the cell is an organelle. And we have smooth ER and rough ER. And again, the smooth ER does not have ribosomes on it, but it's a site of where fatty acid and phospholipid synthesis takes place. But the rough endoplasmic reticulum is studded with ribosomes, and they're pretty much the powerhouse, or not really the powerhouse, that would be the mitochondria that would provide energy. But really it's the factory. The rough endoplasmic reticulum is more like a factory. That's where all your proteins are generated. So the rough endoplasmic reticulum with all those ribosomes on them are constantly churning out new proteins or replenishing proteins for the body. So it's the site of protein synthesis, whereas smooth endoplasmic reticulum without the ribosomes is the site of fatty acid synthesis and phospholipid synthesis. So here's your mitochondria. That's your powerhouse. That's where you get all your energy. And it's the site of oxidative phases of cell respiration. And cell respiration, the Krebs cycle, is what generates ATP. And then we have centrioles are also an organelle within the cell or inside the cell, and the centrioles are found in the chromosome or centrosome of animal and plant cells. They organize the spindle fibers for movement of chromosomes during mitosis and meiosis. So they're going to have a really big part in the rest of our lecture. And the centrioles are made up of microtubules, so they're structural components too. And then our chromosomes exist in homologous pairs in a diploid organism. So diploid is two. Ploidy, if we just looked at the term, if we just looked at the word ploidy, that represents typically the number of organisms. So diploid is two, haploid is one. We'll see ploidy coming back when we talk about mutations later in the semester. So chromosomes exist in homologous pairs in diploid organisms. So homologous pairs are doubling right there. The two per set or two per pair. The homologs are similar but not identical. We saw that in chapter 1 when we looked at the karyotype, and we saw that chromosomes one or each are about the same. Two had the arches a little bit different, but they were still the same. The X chromosome and the Y chromosome look vastly different. They're not homologous, but we see that homologous chromosomes are very similar, just not identical, and within the humans, all of our chromosomes except the sex chromosomes are homologous, meaning that they're all not identical but similar. All of our chromosomes carry genes for the same inherited characteristics. So this chromosomes 1 are identical, are very similar but not identical, but they have the same inherited characteristics on it. They may carry different versions of the same gene, and those versions of the gene are alleles. So maybe over here you have red eyes, and over here you have green eyes, instead of red and green being on the same. But again, that's not really a major difference. That would still be considered carrying the same inherited characteristics. And then our centromere is the constricted region on the chromosome. So that's the little knob when we saw our first chromosome. When we saw the one chromosome where we had the chromosome and then it had a little knob down there and then a little bit more. So that's the centromere, and it's a constricted region on the chromosome, and the location establishes what the chromosome looks like. So the centromere is not always in the exact same spot. So when we saw that karyotype of our chromosomes, we saw that the centromere, the center part, or where they combine or attach the restricted part, is different in each set. And there's four names for those chromosomes: metacentric, submetacentric, acrocentric, and telocentric. We'll get into each of those in the next couple of slides. But another thing to remember before we move on there is sister chromatids with the centromere have a long arm and a short arm. So this would be the short arm. So this is the P arm, and the long arm is the Q arm. And so that's really important to remember as we move forward. So here's our centromere, a little bit more about our centromere. So when it's right in the middle, when the centromere is right in the middle, right there, then it's called then the designation for that pair of chromosomes would be that this pair is metacentric. And then when the centromere is located between the middle and the end of a chromosome, then it's submetacentric. So metacentric's right in the middle. Submetacentric would be near the top or near the bottom. And then acrocentric is right when it's really, really close to the end. It just basically makes it look like a little wishbone. So visually, you can tell the difference between submetacentric and acrocentric. So submetacentric means it's off center a little bit, but it's not way towards the end, whereas acrocentric is right towards the end. And then telocentric is at the end. It's right at the end. So there you do get a little wishbone look. You get the little wishbone part there. But here, this one's a little wishbone with some antlers on it. So there are four designations for chromosomes. And the thing that you want to look at here in the anaphase phase of mitosis, the migration when they separate out, when they separate out. When the chromosomes separate, seen in the first part with metacentric, they're equal pairs, but you get the separation here now, where you get the little arm and the leg moving off. They just separate right there, and you get like right across the middle here. And the same thing up here. You get each chromosome with that little antler sticking out, and then here it just looks like two bones. Kind of looks like your clavicle in a way. And it kind of looks like that part right there. So here's the joint. If you were to think of that as your clavicle, that's the telocentric representation of the chromosomes when they're going through the anaphase part of cell division. So homologous chromosomes, the somatic cells are your body cells. They're not the sex cells. So somatic cells are your muscle cells and liver cells, bone cells, pretty much every cell except the sex cells. And the somatic cells are homologous pairs of chromosomes. So we have 46 chromosomes total, which means we have 23 homologous pairs. So that's our diploid number, our 2n. So 2 times 23 is 46. So again, our n number is 23. And the exceptions are bacteria and viruses have one chromosome, so they don't have a pair. And here's our karyotype again. So this illustrates the physical appearance of the different pairs of homologs. So one thing you want to do is go through this karyotype and figure out which ones belong in this phase up here. So our designation, go through the karyotype and write down the designation of each of those of each of these chromosomes. Sorry, I'm covering the picture here. Here we go. So two different ways of looking at the karyotype. We have the nice picture here with the black and blue and the red, and then we have another one right here, the gray pair. So The difference, the big difference is the sex cells right there. So you have X and Y together, whereas on this one they have X by itself and Y by itself. But they grouped it with visually similar chromosomes. So the X chromosome looks a lot like 4, 5, and 6, and the Y chromosome looks a lot like 21 and 22. Not so much 20, but mostly 21 and 22. So they grouped. So in the gray representation, they're grouped. They're grouped more by visual appearance, and whereas in the colorful one, they're grouped by association. So X and Y always go together, just like the two 22s go together and the two 21s together. And so karyotype for trisomy 21 is 3 chromosome 21s. So you would have another one right there added to it. So what we want to take a look at now, we want to just move along here to our next slide here. So our homologous chromosomes, meiosis converts the diploid number of chromosomes into a haploid number. So meiosis converts the diploid number of chromosomes into the haploid number. And then here's a genome, basically is your genetic information and a haploid set. So here's a bunch of chromosomes or haploid number of chromosomes in a variety of organisms. So we see dogs have 39 haploid is their haploid number, but the fruit fly just 4. Human is 23. So you can go through that just to see an idea of just for comparative purposes. Then our homologous chromosomes contain the identical gene sites along their lengths. So each site on a chromosome is the loci. So when we looked at that picture of the chromosome, and it had its little centromere over here, and we saw on there, it was the one from chapter 1 where there was eye color, like red eyes and green eyes, and there was all different markers on there. Well, each of those spots is considered a locus. Each site is called a locus, and that locus has genes on it that are responsible, obviously, for some kind of functionality. And more details on these are we'll see them in the chromosome bands in a little bit. But one thing I want to mention before we go into the other slides is so one thing I want to mention though again is that the shorter arm by convention is, so that would be right here, this shorter arm right there. Typically the shorter arm is shown above the centromere, and it's the P arm, and the P stands for petite. And then the longer arm is below the centromere, and it's the Q arm. And that was just because Q is the next letter. P was for petite, and then the other letter just was the letter that comes after P in the alphabet. So again, when you draw your chromosomes, you want your short arm or petite arm near the top, and then your centromere, and then the long arm after that. And then biparental inheritance means that you inherit something from inheritance from two parents. So that's what we are. We get one set of chromosomes from one parent, the other set of chromosomes from our other parent. So diploid organisms contain 2 copies of each gene. Again, one from your mother, one from your father. And then our alleles are alternative forms of the same gene. So here's where the text starts using alternative form. Earlier in chapter 1, we said it was all forms of the allele, an allele is all forms of that gene, all the different forms that it could take on. So alternative, remember that alternative, the key thing here is that alternative is the whole entire set of alleles. It's not like there's one that's standard and then you got a whole bunch that are different. You got a whole collection of alleles. Really none takes priority other than when you look at the individual itself. When you're looking at each person, you see which one's dominant and which one's recessive, or which one's expressed and which one is not expressed. So it's different in every person. So the key thing to remember again is that even though we're saying alternative forms, it's really all forms. Alleles are all forms of the same gene that can exist. So here's our chromosome bands, and oh, that picture didn't turn out too good. So our chromosome bands are divided into regions or cytogenetic bands that can be seen using a microscope, along with special stains too, different colored stains. So the bands are labeled. So the petite or short arm, you start at the centromere. So if we drew our chromosome right here, so you start at the P arm and you start at the centromere and you work upwards. So when you're real close to the centromere, that's one. And then you go to the Q arm and there's one there also. That would be Q1, Q2, etc. So you have P1 through 3 or however many you have, and Q1 through however many you have. And you count from the centromere out towards the telomeres. And remember that the telomeres are the little end caps on each chromosome. So at higher resolutions, the subbands can be, you can also see subbands within each of these bands. And the subbands are also numbered from the centromere out towards the telomere. So an example is if you look at the BRCA2 gene, it's on the chromosome 13. So that's this one right here, chromosome 13. It's on the Q arm, the long arm, and it's at loci 12.3. So it indicates it indicates again chromosome 13, the Q arm, and then the 12.3 is band 1, subband 2, and subband 3. So the bolded part is often combined in just saying position 12.3. And then the ends of the chromosomes are telomeres, and they're also labeled, and they're labeled by the pound sign or the number of the chromosome, followed by P-tel or Q-tel. So 7Q-tel indicates 7th chromosome. The long arm or the telomere refers to the end of the long arm of chromosome 7, or the telomere of the long arm of chromosome 7. So here's a better picture of our chromosome bands. So here's our BRCA2 gene as located at 13Q 12.3 right there, and so this is the gene located on chromosome 13, long arm, position 12.3 or 12.3. So changes in BRCA2 gene are often responsible for increased risk of breast cancer, prostate cancer, ovarian cancer, and melanoma. So here's a good picture of what telomeres look like, and here we have in this slide in general, we have a pair of chromosomes, and so they're two chromatids or two sister chromatids. The chromatin is the macromolecules found in the cell that consists of our DNA, proteins, and RNA. And the primary function of chromatin is to package the DNA into a smaller volume to fit within the cell. And then the centromeres, or that central portion again, and they link the sister chromatids. So the centromeres, it's a restricted site, and its job is to pull the two chromatids together, or link the two together. And then our telomeres are at the ends, and they're regions of repetitive nucleotide sequences at each end of the chromatid, and it forms like a little boot or a little knob that protects the end of the chromosome from deterioration. Some recent studies show that as people age, their chromosomes start to shred, or that the telomeres start winding down or unwinding or fraying. And you see that, or they're starting to see that or they're starting to notice or study the effects or what chromosomes look like in older populations or older people. One other thing too is sun damage actually also enhances the ability of the telomere to fray, or I shouldn't say ability of it to fray. Its job is not to fray. It's supposed to be a protective boot there, and it keeps the chromosome from deteriorating. But as people age, the telomeres become weak and they start to fray. But then environmental factors like sun exposure causes the telomeres to fray as well. And that's what's thought when you see a person that spends an extraordinary amount of time in the sun. Oftentimes they look a lot older than they really are. Their skin thickens, it gets a little hardened to look at, maybe gets pretty wrinkled. And that's thought to be from the telomeres fraying. their chromosomes are not as strong anymore, and not as weak, or the cells within your skin are so damaged, and that damage is often associated with frayed ends of telomeres. So your centromere is your sex-determining chromosomes, your centromere is the what ties them together. But the sex-determining chromosomes are not homologous, but they behave as homologs in meiosis, meaning that they pair up together. So an example in humans is the X and the Y chromosomes are where they're the sex chromosomes that are not homologous. So the centromere, though, if we look over here on the X and the Y, the centromere, you might almost see a little ball right there on the top, and then here's the centromere in the X chromosome. So I just covered that up. I erase over that. So you can see the centromere in, you can see, let me see if I can draw that as a fine point. Yes, the centromere you can see right in the center, and then this one will be right near the top. So a little hard to see them on the Y chromosome, but the X chromosome, the centromere, is pulling the two bands together, or two chromosomes together there. So, but they're not homologous though. They just behave as homologs as each part separates out, or each one separates out, and during the cell division. So now for that cell cycle, and particularly mitosis and meiosis. So mitosis partitions chromosomes into dividing cells, and some definitions here is that the zygote is the single-celled fertilized egg from a multicellular organism. So humans are multicellular organisms, and that one fertilized egg is a zygote. And then a karyokinesis is the genetic material that's partitioned the daughter cells during a nuclear division, and that's or nuclear division would be cell division when the nucleus divides. And cytokinesis is the cytoplasmic division that follows. So here's that whole cycle put together. So the cell cycle is a process that occurs a lot over an organism's lifetime. We shouldn't think of cell division as just creating an egg or a sperm. It's far more than that. It's the replenishment of your skin cells. When you exfoliate, you have to replenish those cells, and so new cells are generated. And I'm sure you probably covered that in your anatomy class when you studied skin. But that's really what's happening as those cells move from the bottommost layer. I think there's 7 layers of the skin. They move from the bottom layer to the outermost layer. Well, near the bottom is where the cell division is actually occurring and making new skin cells. But every organ within our body creates new cells, not just new skin cells, but all new cells. Bone cells are regenerating and regrowing each time, or replenishing. So are liver cells. So basically all your cells in your body are constantly replenishing and growing and going through the cell cycle. So the cell cycle is a process that occurs over an organism's lifetime, and its big purpose, aside from regenerating your replenishing sloughed off cells, is that the genetic material is replicated in that cell division or cell process, cell cycle, and it's passed along to the new cells when that division happens. So the stages of the cell cycle are first we start out with growth, and then we have the mitosis or nuclear division, and then cell division. So the phases in the mitotic divisions are the first phase is interphase, and interphase has 3 stages, and we see interphase as G1, where it prepares the DNA and chromosome replication. The S phase is where DNA and the chromosomes replicate. And then G2 is where the cell prepares for division, which occurs in the mitotic phase, the M phase. And the mitotic phase is the one that's further broken down into prophase, metaphase, anaphase, and telophase. So we'll look at each of those in more detail. So here's those stages again. This slide right here is the same one right here, prophase. The only difference is that on this slide here we have pro-metaphase, and that's really right between prophase and metaphase. So we'll get to all these definitions as we go through these charts right here. So here's all of our phases. So here they all are again. So we have a list up here. We have the stages, and then right here we have the stages again. So here's our first stage, interphase, right here. So that's when the chromosomes duplicate, and chromosomes duplicate, and the copies remain attached to each other, though. So you duplicate your, in interphase, you duplicate your chromosomes, but they're still attached. They're still paired up with who they need to be paired with. And then in prophase, in the nucleus, chromosomes are condensing here, and this is the first time that they're visible. So prophase is when prophase is the first time that your chromosomes can actually be visible under a microscope. So it's the second mitotic phase which is prophase. And then here's your prometaphase, and that's where the nuclear membrane is actually breaking apart. And so it's the membrane, it's not the cell membrane that's breaking, it's the nuclear membrane that's breaking. So that's a key difference. You want to remember that the nuclear membrane breaks apart in prometaphase. And that's where the spindles start interacting with the chromosomes. So that's that structural component, and they're going to act with they're going to interact with the chromosomes. So we see them right up in here, and it's kind of like they're pulling them apart, like they're traffic cops, kind of guiding them to where they need to be. So I'm going to erase that so you can see it better. So that yellow structure again, in metaphase, they're the spindles that are interacting with the chromosomes. So they start interacting in prometaphase, and then they continue. The copied chromosomes are aligning in the middle of the spindle. So they're kind of lining up on the equator, and the spindles are guiding them to do so. And then in anaphase, the chromosomes are separating into two genetically identical groups, and they move to the opposite ends of the spindle. So your spindle now created poles. You have a north pole and a south pole with spindles at each end, and then the spindle arms are separating the chromosomes, they're pulling them apart. In metaphase, the spindles made the chromosomes line up along the equator. And then in anaphase, the spindles separated the chromosomes. And then in telophase, here's where the cell, and over here we have the nuclear membrane beginning to disintegrate. Well, here we have the cell membrane now. We have the nuclear membrane forms around each of the two sets of chromosomes. So you got two nuclear membranes there. And then the chromosomes begin to spread out, and the spindles break down. And but your plasma membrane now is starting to wear down, and it's getting ready to actually divide, and that's what you have in cytokinesis right here. You have you have your two daughter cell, or two, not daughter cells. You have your two. Oh yeah, I'm sorry, they are called daughter cells. So your mitotic phase, you still wind up with two daughter cells, and they each have the same number of chromosomes as the parent. And so in humans, such cells, or these daughter cells, have 23 chromosomes, and they're called diploid. So each daughter cell receives a complete homologous set of chromosomes, but each daughter cell receives only one sister chromatid of each chromosome. So you had your divisions up here where you're kind of separating them out, where you duplicate it and then you separated them. So interphase, here's some pictures of slides of what we normally see in our drawings versus this is what we see in our drawings, and then this is what we see through a microscope. So if you looked at cell division or mitotic phases, the interphase in particular under a microscope, here's what it would look like, and it's where the chromosomes are extended and uncoiled, forming the chromatin, and that's your interphase part. And then prophase and your sister chromatids. Prophase, the centrioles divide and they move and they start establishing poles. And the nuclear envelope breaks down and chromosomes are now becoming visible. And the sister chromatids are the two parts of each chromosome connected at the centromere. They're held together by multiple subunit proteins called a cohesion protein. And then you have prometaphase, where it's the period of the chromosome movement towards the equators now. So you're going to have them line up along the equator, or that. So you're going to have them start lining. They're going to start lining up this way right here, because here's your pole There's your poles, and you're going to line up along the equator. And that's what's starting to happen right here. They're lining up along the equator in prometaphase. And then in metaphase, the chromosome configuration looks nice and neat along that, nice and neat along the equator. And the centromeres align on the metaphase plate, and then we're going to start seeing them pull apart in the next one. So in metaphase, kinetochore is the disc-shaped protein structure located on the centromere, and the spindle fibers are binding to that kinetochore to pull the sister chromatids apart. And then the chromosomes start migrating towards the poles. And the kinetochore microtubules have one end near the centromere region or centrosome region, and the other one anchored at the kinetochore region. And then a cohesion protein complex holds the chromatids together. Separase is the enzyme that degrades the cohesion, allowing for them to be pulled apart. And then sugoshin is the protein that protects cohesion from being degraded by separase. So it doesn't actually. Sugoshin is a protein that's meant to protect the cohesion from being degraded, but obviously during cell division you don't want it to work, so it turns off. There's some regulatory mechanisms that allow it or that help it to turn off, so that the degradation of cohesion occurs, and then you can get the separation or the independent assortment of the chromosomes in the metaphase part. So here's a picture what that looks like, or sister chromatids, cohesion holding them together, and then the centromere region, your spindle fibers connecting on. One end is the kinetochore, and then the upper end is the microtubules, and then of course it pulls apart. So here's your sugoshin sitting on here, but it turns itself, a regulatory mechanism as it turns off during cell division to allow the chromosomes to be separated. And then anaphase is the disjunction. That's when they are pulled apart and officially pulled apart. So the sister chromatids separate and into daughter chromosomes, and the daughter chromosomes migrate to the opposite poles by shortening of the fibers, spindle fibers. And then in telophase is where cytokinesis is a part of that. It's the part of the cell division when the cytoplasm of a single cell divides into two daughter cells. And then the uncoiling of the chromosomes happen, and so they get loose again like they were in the very beginning, and the nuclear envelope reforms around them. Spindle fibers disappear, they go away, now they're not needed anymore, and the nuclear envelope forms around the nucleus again, or reforms. And then you wind up with two separate daughter cells. So here's some more microscope pictures lined up with the graphic pictures or simulation pictures. Same thing here. So mitosis partitions chromosomes into dividing cells, and it produces the daughter cells with the full diploid complement of chromosomes. The animation I have in a separate file. I'm going to try and post that on Canvas. So cell cycle regulation and checkpoints, there's a lot of regulation, a lot of regulatory events within mitosis, and a lot of checkpoints. So basically, there's a checkpoint at every phase. So many mutations do occur though, and like the whole point of cell cycle regulation and checkpoint regulation is to prevent mutations and prevent things from going wrong. But it happens though. Mutations are going to occur, and they're not going to get caught. They're going to get sent through, and we'll talk about some when we get into the chapter on mutations. We'll go into greater detail about which mutations are lethal and which ones pass through. An obvious one that passes through is Down syndrome. That's trisomy 21. So that's a mutation, but it's not synonymous with cell death. So consequently, the zygote or the growing child continues to develop, even though there's a mutation had occurred. So cell division mutations, enzymes called kinases typically are trying to prevent the mutations. It can add a phosphate to another protein to regulate the cell cycle. And typically, the kinases or enzymes are master control molecules functioning in conjunction with cyclin proteins. And all the checkpoints are to monitor mitosis for error. And sometimes the errors are insurmountable, so apoptosis or cell death occurs. Other times they're not insurmountable, and you end up with either during prior to birth, it would be a congenital defect that would occur because of a mutation. But then once you're born, mutations like that occur because of sun damage could create a tumor to start growing, skin cancer. Same thing with mutations of a gene that causes breast cancer or colon cancer. Basically, any of the cancers are because some regulatory mechanism didn't catch the prolific cell growth. So we finished up mitosis, and now here we are in meiosis. Meiosis, it's the reduction of genetic content and chromosomes is basically what happens here. Meiosis reduces the genetic content. So it's a mechanism by which the diploid amount of genetic information gets reduced into the haploid amount. So you go from 46 to 23, and 23 individual chromosomes, not pairs. So it produces haploid gametes, each containing one member of a homologous pair of chromosomes. And here's where the genetic variation is going to occur, because you're getting one chromosome from your mom and one from your dad. And then when you have a child, then one of your chromosomes gets passed down, but that one chromosome has information from one of your parents on it. So a lot of variation, genetic variation occurs in meiosis. And it's also the gamete, again, a definition is the ovum or egg or the sperm. So a gamete is one or the other, either the egg or the sperm. And our little Billy box down there says meiosis is critical to successful sexual reproduction of all diploid organisms. That's probably the best way to describe meiosis. It's definitely critical to this sexual reproduction of all diploid organisms because it's producing those gametes. So meiotic event is crossing over, and it's where genetic exchange between members of homologous pairs of chromosomes occurs. So when one part of a chromosome crosses over or changes with another, the genetic material crosses over. And then part of those meiotic events, meiosis 1 and 2, 1 is the reductional division, and then 2 is when they equate back together. So DNA synthesis occurs during interphase before the beginning of meiosis 1, and then does not occur again before meiosis 2. So meiosis significantly increases the level of genetic variation via crossover. And we'll see some of those pictures later on in another lecture. And then here are meiosis 1 and 2, similarities. They each have prophase, metaphase, anaphase, and telophase stages. So similarities between the two meiosis stages. But then there's also similarity between meiosis and mitosis. So you can see the same set of phases where you get your daughter cells. But here's where the reductional division down here is occurring. So we don't see that in mitosis. And then, of course, mitosis gives you 2 daughter cells, but meiosis gives you four of the gametes. I'll read that off here. So you get your gametes at the end of mitosis. At the end of meiosis 1 is where you get your gametes. And then meiosis, when we look at what gets segregated in meiosis 1 and meiosis 2. So these are some of the things you could think about as you're reading through the lecture to make sure you know what goes on in each stage. And then which ones are more similar. You can make the comparison here. And then, of course, meiosis generates genetic variation by passing off one chromosome from each parent. So the development of gametes, the development varies between spermatogenesis for the sperm and oogenesis for the egg. And then the male gametes are produced again by spermatogenesis in the testes, and female gametes are produced by oogenesis in the ovary. So primary spermatocyte undergoes meiosis 1 and gives us 2 secondary spermatocytes, which then undergo meiosis 2 to give us 4 haploid spermatids. And then oogenesis is the formation of ova in the ovaries, and they give results in 4 daughter cells that do not receive equal cytoplasm. Only one daughter cell receives the cytoplasm, and it's as the primary polar body, and it undergoes meiosis 1 and 2 and then develops into the ovum. And then our chromosomes, electron microscopy is what reveals the physical nature as what we're able to see under a microscope. Using an electron microscope, we can see the chromosomes pretty well, pretty clearly, and we can see each meiotic division. You can look at a regular or through kind of cheap microscopes and still see a dividing cell. There's a lot of I think Flynn or Fisher sells slides, microscope slides that already stained and include copies of chromosomes or copies of each phase of cell division. And so that's what those earlier slides were that we saw. Just a regular plate with the cells actually dividing. But the electron microscope gives a greater level of detail. That's what this one, the second part is right there. You can really see the chromosomes pretty good. And then you can zoom in on certain pairs and see more particular spots if you'd like. So that actually is everything. I didn't put my questions slide on there, but of course, if you have any questions at all, please don't hesitate to contact me.